Okay, well, good morning, everyone, and welcome to the GCC Single Cell Omics Symposium. We have over 300 people registered for the meeting, so I'm very excited to have everyone participating. To give a little bit of background, the GCC, or Gulf Coast Consortium, is an organization funded by the Keck Foundation. Okay, well, good morning, everyone. Oh, sorry about that. That was a little bit of echo there from the other umbrella platform. So just to give a little bit of background on GCC and the Keck Foundation again, it's an organization that tries to bring together investigators here in the Texas Medical Center from all the different universities, from MD Anderson, Baylor, Rice, University of Houston, UTMB, and others that are interested in single cell technologies, both computational approaches, technologies, and biological applications. And so we have a weekly seminar series, we have networking events, and then this is our annual symposium that we'll be holding every year for the next number of years. We have a very exciting program today. Our first keynote speaker will be Stephen Quake from Stanford University. And then our second keynote will be Ort Rosenblatt from MIT Broad, and she'll be talking later today at 2 o'clock. The conference is organized into the 3 different sessions. The first one is single cells in cancer research. The second one will be single cell genomic applications. And the third one will be computational approaches, software tools, and technologies. We'll be using the umbrella platform, so you can stream all the talks there. If you want to ask questions, please use the chat window and insert your question there at any point during the talk, and then we're going to select those to ask the speakers. There will be 3 vendors talking: 10x Genomics, Illumina, and Protein Simple from 11:10 to 11:40 today. And we want to thank everyone for the funding support, both the vendors, Rice University, GCC, and others. And there'll be poster sessions too, 23 posters in total. That will be around lunchtime, so from 12:50 to 2 p.m. And there'll also be vendor workshops if you want to attend those during that time. Okay, so welcome back, everyone. We're now going to start the first session of the symposium. And this one's on single cells in cancer research. And we have 3 speakers in this session. So Koichi Takahashi from MD Anderson, Fang Wang, also from MD Anderson, and myself. And we're going to start with a talk by Koichi Takahashi. Koichi is a physician scientist and really, I would say, one of the first or early people taking single cell sequencing technologies and moving them into clinical applications. He's in the Department of Leukemia, and he's also in the Department of Genomic Medicine. He did his clinical training previously and his postdoc with Andy Futriol in the Department of Genomic Medicine. And without further ado, I'm going to allow him to take over and give his talk, which is titled Single Cell Genomics Reveals Clonal Evolution in AML. So thank you.
Thank you so much, Nick. I hope my slides can be seen. Slides are okay? Okay. Okay, great. Sorry about that. And thank you so much, Nick. My name is Koichi Takahashi. I'm the physician scientist in leukemia department and genomic medicine department at MD Anderson. And it's really a pleasure to actually be at my office and able to present this in this symposium. Our lab has been interested in to profile clonal heterogeneity of tumor from a variety of reasons, because precise mapping of heterogeneity really allows us to understand a lot of important biology about the tumor development. One is the genetic instability of how each cell acquire mutations, and how the tumor ecosystem or the selective pressure in the tumor ecosystem shapes the architecture of the clonal heterogeneity. And also precise mapping of heterogeneity or diversity allows us to infer how the tumor evolved from the beginning. So inferring the history of the tumor evolution. That is also largely depends on how precise you map the heterogeneity. And from the clinical standpoint, describing the heterogeneity precisely will allow us to understand what is the resistant clone or identify relapse-fated clones in the tumor, and also understand the prognosis of those tumors. So from those reasons, our lab has been heavily involved or heavily interested in to analyze the heterogeneity of tumors using the state-of-the-art technology such as single cells. So how can we describe the clonal heterogeneity of diversity accurately? Historically, we have been relying on the bulk sequencing technologies to infer the clonal heterogeneity using this variant allele frequency hierarchy in the mutations. So this paper in the Nature Communication really illustrates this, the capability and also at the same time the limitation of the bulk sequencing to infer the clonal architecture well. So when you have 10 mutations with the variant allele frequency and you group them based on the variant allele frequency, and you try to create a model of clonal architecture. But if you only have this snapshot of bulk sequencing data, there's multiple possible models of clonal architecture. One is this linear clonal architecture, and the other possibility is this branching models. And bulk sequencing data, unfortunately, if you only have a one-time snapshot samples, unable to really determine which model is the correct one. If you have multi-region sequencing, so multiple biopsies from multiple location of the tumors, or if you also have a longitudinal samples and track the mutation, how they travel together, these analysis will definitely help understanding the clonal heterogeneity better. But a lot of times getting multi-region samples or longitudinal sample is costly, and also it's labor intensive. So this is not performed routinely. So as everyone knows, Nick has pioneered this field of genotyping each single cell from the tumor. He has done this in the breast cancer in 2011 and really described the copy number, single cell copy number profiles extensively in the breast cancer tumor samples. And he has done that also in the point mutations spectrum as well. But accessing DNA in the single cell is technically challenging. Unlike RNA, where multiple copies exist in the cytoplasm and it's easily accessible, in theory, in each single cell, there are only 2 copies of DNA, and those are tightly compacted in the nucleus and chromatin. So technically, it has been very difficult to access the DNA at the single cell level, and that is, there's no surprise that single cell DNA sequencing commercialization has been lagging behind the single cell RNA sequencing technologies. And also, in order to do the whole genome level single cell profiling, we still need to use this whole genome amplification process, which is associated with introduction of a lot of false positives and also the false negatives. That is very nicely summarized by Nick in this review. So from that context, our lab was fortunate enough to collaborate or get involved with the development of this high throughput single cell targeted DNA sequencing platform called, now it's commercialized and named Tapestry. This uses a microfluidic platform to singularize a single cell and perform PCR-based amplicon deep sequencing in each single cell. And I want to emphasize that this is not a single cell whole exome or whole genome sequencing platform. It is a single cell targeted DNA sequencing platform, but nonetheless, it allows us to genotype up to 500 amplicons. And what is most important for us was that the throughputness, that it could genotype roughly 5,000 to 7,000 cells in a run. And we had a pilot analysis together with Mission Bio using our acute myeloid leukemia samples, and that remarkably showed a nice clonal heterogeneity map of that samples. And we quickly moved on to expand this effort to the large scale single cell genotyping effort of acute myeloid leukemia samples. So for those of you who are not familiar with acute myeloid leukemia, it's clonal hematopoietic malignancies. And the reason why acute myeloid leukemia was, or we chose this as a first tumor to profile the single cell genotyping was that not only that I work in the leukemia department, but also the 90% of AML carry at least one driver mutation. And this driver mutation spectrum has been extensively characterized by the bulk sequencing efforts such as TCGA or ICGC. And each sample contains median about 3 driver mutations. And also the leukemia is a liquid tumor where we can easily access the tumor sample by doing bone marrow aspiration, or in some case, peripheral blood can be used to access the samples. And so therefore, we undertook this effort in acute myeloid leukemia to genotype by single cell technology. So far, we have profiled about 150 bone marrow samples taken from 123 AML patients. And we use this tapestry platform to profile the single cell level clonal diversity. We used the 2 panels. One was the pre-designed panel by Mission Bio, which covers 19 genes with the 50 amplicons. But we also developed our own custom panel, which consists of 290 amplicons covering 38 driver genes. And this custom panel should cover most of the driver genes that have been identified in AML by the previous bulk sequencing effort. So this analysis is not a discovery analysis to identify any new novel driver mutation, but using the previous knowledge of the driver mutation and profile that at the single cell level in acute myeloid leukemia to really understand the clonal diversity well. This experiment was done primarily by Dr. Morita, one of the graduate students in my lab, and was helped by Tanaka and Sasaki, our postdocs, and bioinformatic process was mainly done by Feng Wang in our lab. And all of the samples were concurrently analyzed by bulk next-gen sequencing or ddPCR in order to orthogonally validate the mutations that was detected by the tapestry. And also in some cases, we've done a SNP array to validate the zygosity of the mutations. Since this is a new, relatively new platform, I'm gonna start with the QC metrics of these platform, but this is as I said, it's a PCR amplicon-based sequencing. So depending on the amplicons, there are good performers and the bad performers. And most of the bad performers are covering the genes where it's a GC-rich region. So some of the genes, for example, SRSF2 or RUNX1 or P53, where those regions are GC-rich, they had a poor coverage. But overall, a median, we cover 24x coverage per cell, per amplicon, which we think it's a pretty sufficient to genotype accurately for each single cell, because each single cell has theoretically, again, 2 copies. And if you do 24x, you have a chance to cover both alleles at least 10 times in theory. We input 270,000 cells into the machine, and the capture rate, at least at this point, or when we wrote the paper, it was about 3%. And I think the platform is improving the capture rate to now about 10%. Although we are losing 97% of the cells during the processing, at least we get information of the genotype information on about 7,000 cells or 10,000 cells in one sample, which we think was pretty good enough to get a statistical power to understand the relationship between the driver mutations. And although we're losing 97% of the cells, when we compare the variant allele frequency from the bulk sequencing and the single cell derived variant allele frequency, there's a nice concordance between the two, suggesting that there's really no bias in this cell sequencing process. So although we are analyzing 3% of the input cells, it is really representing the entire bulk tumor cells. We estimated the allele dropout based on the SNP row size, and the median was anywhere around 8.7%, and it never exceeded 20% in our samples. This is the comparison of mutation detection between the bulk sequencing and the single cell DNA sequencing. And all of the mutations are, again, orthogonally validated. And what was reassuring was that 100% of what we detected in the bulk sequencing was also detected by the single cell DNA sequencing. And in fact, single cell DNA sequencing detected more mutations than the bulk sequencing, particularly in FLT3 gene, especially this internal tandem duplication mutation in FLT3 gene. And this internal tandem duplication mutation has been notoriously difficult to detect using the conventional bulk sequencing technologies. So this was really gratifying to see that the single cell DNA sequencing using this PCR amplicon-based technologies really detect a lot of FLT3 ITD mutations. We also, by the dilution assay, we estimated the limit of detection of this platform to be about 0.1% of the cellular population. In our samples, we detected median 4 subclones based on this single cell profiling. There was modest correlation between the clonal diversity of each sample and the age of that samples, which I think Steve also mentioned in the previous presentation. So the more aged the patient is, we saw that increase in this clonal diversity detected by this platform. Obviously, there is a linear correlation between the number of mutation we detected and also the increase in the clonal diversity that was measured by the Shannon index. But the more mutation in individual clone did not necessarily contribute to fitness per se, which was measured by this cancer cell fraction of the mutations. So there was maybe some linear kind of correlation until the 3 driver mutations in individual clones with correlation with the fitness. However, after more than 3 mutation, having 4 or 5 or 6 or 7, 8 mutations in the clone did not necessarily contribute to the increase in the size of that clone. We also analyzed the correlation between the mutation and the clonal diversity. And we saw that having RAS mutation or signaling type mutations was correlated with the higher diversity in that samples. And having a P53 mutation or DNA damage actually response mutation was negatively correlated with the Shannon index, at least in our cohort. For each sample, we get this type of map of single cell genotyping. Here, each column represents each cell and each row represents each mutation. And if it's blue colored, it's mutated, and it's white colored, it's wild type. And the gray means that we could not conclusively determine the genotype. We started to observe quite interesting mutual exclusivity patterns between functionally redundant oncogenic mutations. For example, in this case, where the sample had a concurrent IDH1 and IDH2 mutation, these 2 mutations were clearly mutually exclusive to each other at the cell level. And this was also similar in this case where this patient had 2 types of FLT3 mutations. One was internal tandem duplication and other one was D835Y clone. And they were nicely mutually exclusive at the cellular level. And we summarized this mutation co-occurrence and mutual exclusivity at the cell level based on our analysis, which largely recapitulated what we have known by the bulk sequencing data, but nonetheless, I think validating this at the cellular level by the single cell sequencing was quite gratifying. So next, using this single cell genotyping data, we tried to infer or reconstruct the evolutionary history of the AML. So using this single cell genotyping data, we wanted to infer or estimate how this tumor was developed and in what mutation order. For that, we collaborate with Nico Bierenwinkel in ETH Zurich, where they have developed this algorithm called SITE, which was specifically designed to reconstruct the mutation order or phylogeny using the single cell sequencing data. And using this data, we reconstructed our entire samples, and about half of them, our AML sample had this linear mutation structure, that the driver mutation was kind of linearly or stepwise acquired. But other half, we observed this branching evolution pattern. Here, I'm showing one representative case which had a very complicated branching structures of the mutations. And among this branching case, there were 3 cases where we identified this convergent evolution pattern in acute myeloid leukemia. This case is a very fascinating case where and I will emphasize that this sample was collected at the time of diagnosis, so without any selective pressure given from the therapy, that this sample had this almost 11 clones that was parallel, what was evolving in parallel. However, they ended up acquiring very similar functional mutation in the end. It started with NPM1 mutation and acquired next as IDH1 or either IDH2. And in the end, they acquired this signaling mutation that is in this RAS or RTK pathways. So why does this tumor needed to have 11 parallel subclones ended up having essentially similar functional mutation? And there must be some selective pressure in this patient's tumor ecosystem, or in the bone marrow ecosystem in this case, that favors this configuration of the clonal structure. And this really generates a hypothesis: what is this selective pressure that is unique to this patient that configure this clonal architecture? Convergent evolution itself is not uniquely identified in AML, and it has been previously identified in renal cell carcinoma by multi-region sequencing. But our results, I think, was really the first to describe this in acute myeloid leukemia at the single cell level. Next, this single cell sequencing was done in the snapshot of the samples, but is this diversity that we are observing, is it just a snapshot, or is this really supported by the diverse stem cell population? And in order to test that, we have transplanted these samples into NGS mice and let it engraft and then analyze the engrafted samples with the same single cell platform. And for this representative case that I'm showing, we have see almost exact clonal architecture in engrafted samples, suggesting that each of these parallel subclone is supported by its own leukemia initiating or leukemia stem cells clones. By virtue of time, I'm going to skip other examples, but here I wanted to show the similar example of that. Next, we asked whether the mutation order that we inferred from the single cell data is clinically relevant or not. Because we have known from the bulk sequencing study that what mutation occurred first or mutation order could affect the clinical phenotype. I'm citing this New England Journal paper by Ortman, where they analyzed this, they identified this TET2 first case or JAK2 mutation first case among the myeloproliferative neoplasms. And they show that depending on what mutation they started with, the prognosis is different and their clinical characteristics is different. So using our mutation inference from the single cell data, we analyze whether that affects any clinical phenotype or relevance. We summarize the initiating mutation in our case, and in AML, initiating mutation largely involve these epigenetic mutations that is working in the DNA methylation or chromatin structures. And signaling mutations like RAS or FLT3 mutations are rarely acquired as a first mutation. However, there are rare cases which started with these signaling mutations. And when we compare the clinical phenotype of these, for example, this RAS first cases, that is rare, but we observed, these cases had increase in the monocyte counts in both bone marrow and peripheral blood blood, which suggests that RAS first AML might be associated with this monocytic phenotype. This is broadly consistent with the previously known knowledge that in monocytic leukemia, for example, this chronic myelomonocytic leukemia, RAS mutation has been shown to be acquired first. So these data kind of suggest that there is some relevancy between mutation order and the clinical phenotype. So to explore that further, we did do the single cell simultaneous DNA and protein profiling of this in some of the cases, and we correlated the mutation with their cell surface epitope. And we did confirm that certain mutation has this specific correlation with the cell surface epitope. And using this simultaneous platform, we also show that how genotypic evolution correlated with the phenotypic shift during this AML development, where in this case was started with TET2 and subsequently acquired these mutation. And gradually, during this genetic transition, we saw this multilineage phenotype toward the myeloid skewed phenotype, and finally acquired this blastic phenotype.
Koichi, I'm sorry, you're out of time.
Okay, I'm going to finish here. I will skip these longitudinal cases, but just in summary, I wanted to show this the capability of this single cell sequencing in order to profile this clonal diversity at the single cell level. And there is this genetic phenotypic correlation that we found, and I couldn't go over this longitudinal analysis, but we apply this in the longitudinal samples to really understand the treatment response at the individual clonal level. And I really want to thank all the collaborators and also Mission Bio folks in helping us completing this project. And this paper is in press at the Nature Communications, so I really encourage you to read this paper when it gets published. Thank you so much, and sorry that I exceeded a little time.
Okay, well, thank you so much, Koichi. Unfortunately, we are well over time here. So I think if anyone has questions, please post them into the stream channel umbrella. And I think Koichi can just see the questions there and respond to you either by email or through the umbrella system. Okay, thank you very much, Koichi. That was a wonderful talk.
Absolutely. Thank you, Nick.
Okay, so now we're going to switch it over to the last talk today. And actually, Fang Wang is in China right now, and she's having trouble being able to give this talk. So her mentor, Ken Chen, is here, and he's going to fill in and give the talk. Ken is an associate professor here at MD Anderson in the bioinformatics department. And his talk today is called Aneuploidy Lineage Tracing, Enabling Cancer Gene Discovery and Single Cell Sequencing Data. So I'll hand it over to you, Ken.
Great, thank you. Can you hear me?
Yes, sounds good.
Okay, great. Okay, so as Nick introduced, I'm presenting on behalf of Fang, who was a postdoc and is now a faculty position in China. And so just before getting to the subject today, a quick description of the lab. So I'm running a lab of computational biology at the MD Anderson Cancer Center. And the lab actually have a pretty diverse interest and in the different area of informatics, starting with genomics, structural variation, and much more get into multi-omics, including not just genomics, but also transcriptomics and nowadays. And we're interested in evolution at both bulk and single cell level. And we're also interested in functionalize genomics and transcriptomics in the context of molecular medicine, both targeted therapy, as well increasingly more of immunotherapy, particularly the chimeric antigen receptor-based targeted immunotherapy. So, so the overarching problem we're trying to tackle, I guess I thank Koichi and Nick for a very nice overview. We're trying to computationally infer or characterize this evolutionary process of cancer from very early stage, before initiation, all the way up to the late stage of cancer, what's happening genomically and microenvironment. So, but we focus very much on the computationally challenging issue in this process, which included some I described and Nick and others described before. One of the particular problem of today is how do we categorize the order of the evolution and which events are important, potentially functionally important. So I just like to quickly show two recent programs I think is pretty important. To understand this evolution, one important thing is to do multi-omics, is to trying to integrate different modality, not just genomics, potentially expression, content accessibility, and also protein surface marker and potentially spatial transcriptomics together. So because the modality are produced by different assays at this point, so one important task is how do we integrate them together in an unbiased way? So there's a new problem developed by a mathematician in my group that perform this unbiased integration of different modality and trying to acquire the modern omics profile for each single cell and trying to infer the regulatory mechanism, for example. So welcome to check it out. Another important aspect is how do we identify important genes that is differential, that is reflecting some of the function. One of the limitations of the current paradigm is that they are so clustering dependent. Depending on the clustering results and parameters, you could have different set of cells to perform the analysis. So we're transitioning into more of a cluster agnostic independent process to detect the gradient. So this is a program developed by Xiaoheng Liang, a student in Rice Computer Science Department. And it was able to actually capture a lot of not only cell lineage markers, but also potentially functional pre-resistance, drug resistance markers by just trying to convert the high dimensional data into a low dimensional data and trying to identify the important genes and markers in this process. So this is also available in GitHub. Feel free to check it out and follow up with them. Okay, back to today's subject. I think the core we're trying to do, at least partially, is to follow this model proposed already by Peter Novell. I think Nick referenced to this into more detailed model. We're more thinking in terms of a gradual evolutionary model process. Previously, we have done this using single nucleotide polymorphism, single nucleotide variants from the single cell data by trying to expanding the existing phylogenetics algorithm in order to model parallel evolution. So we allow mutation to occur multiple times in different cells in the same evolutionary process, and we allow handling of different sequence and variant calling error, including dropout, in this statistical probabilistic model with collaboration with Nick Luai and Hamin Zafar from Rice, all published work. So, however, the other important aspect of tumor evolution is the copy number, which is potentially a driver in itself, but also condition the genomes to more other mutations, and also a mechanism, functional mechanism, for example, immune evasion in the process. So therefore, it's also important to be able to examine the order and also the process of how copy number evolve at the single cell level. I think Nick's talk has actually very well summarized the motivation. And I just like to add on, this has a lot of clinical implication. Genome instability has been a target of different therapy. And mainly 88% of actual TCG tumor has arm level aneuploidy. So this is a widespread and clinically important problem. So the data that for copy number evolution can come in from different sources, single cell DNA sequence, and those are relatively outdated. But Nick just showed their recent work on this acoustic, not TACMA-based acoustic methods, which is able to actually profile this at very high throughput in many cells, genome-wide. But you could also get this from single cell RNA sequencing and single cell attack sequencing data. Albeit they're highly noisy. So how do you infer it from this is the subject of our study. So how do we construct the tumor evolution from the single cell copy number profile, integer copy number profile, tens of thousands of cells with potentially 100 KB genome resolution, And how do we discover the events that potentially associate with cell fitness and also lineage expansion based on the structure we obtained, we infer from the genome. So as you know, this is complicated because copy number can evolve in many different ways. There's a bunch of many mechanism underneath DNA repair, damage repair, DNA replication that could trigger copy number alteration in different type and occur in different order. So how do we solve this is actually a very computational challenging issue. And as you know, copy number alteration also do not overlap. It actually can overlap at the same locus. So this is make it difficult to computationally model this type of thing. And it also have this loss, which cannot be reverted. It's an important phenomenon for copy number alteration. Previously, people are trying to expand phylogenetics models to do this. This is one of the early work. However, one particular issue here is those are all appear to be NP hard issues, so many combinations. So it's impractical to actually solve this using the standard phylogenetics algorithms. They only scale to tons of cells. So therefore, we developed an algorithm called minima event distance aneuploidy lineage tree algorithm, which basically look at those integer copy number profile and try to construct a minimum spanning tree, which reflects the minimum number of single copy gain and loss that could have occurred throughout the evolutionary process. So the algorithm works like this. Basically, you obtain a representation, each circle is a cell, individual cell, arrow representing the direction of evolution, so early to late events, and they weight the thickness of edge representing how many copy number happened between two states. And you could apply this to any, for example, TNB's triple negative breast cancer sample and try to observe the evolutionary process and trying to identify clones based on from the tree, and also label the clinical information on the tree, for example. And one of the tricks is to figure out what are the intermediate steps, single copy events that has actually occurred. So this is an inference issue, but we solve it with a greedy way. So we're trying to identify the longest single copy gain and loss between minimum number, within the minimum number of steps to transition the states from A to B. So this is a very efficient solvable problem. And to construct this into a tree, we have to basically convert the graph into a kind of a directed tree representation, which actually could also be solved by a previous algorithm. And this has a polynomial computational complexity, which make it scalable to thousands of cells, basically. Of course, it's difficult to validate those exactly, but I think we can, validate through some sort of downstream analysis by looking at the tree structure, by looking at events that is associated with a larger clone, recurrent associated with larger clone, especially if they occur, reoccur relative to a background trees, which we random permuted the data generated from. And we can also examine this across multiple samples and see which of those events are reoccurring associated with the clonal expansion process as a way to identify the important events and genes. And established that statistical process, and we perform simulation. This is actually very much similar to the previous work where we randomly generate a tree by kind of spiking different driver genes and expand the cells according to the presence of those driver events. But we're kind of spanning in copy number evolutions under a particular mechanism. And so we can compare with this ground truth, we can compare with other traditional phylogenetics approach as well as the approach applied to the bulk data analysis. And we can observe a very evident performance gain using our approach. And of course, we started to apply this on the real data. So this is one data from Nick's previous clinical trial data under triple negative breast cancer, chemotherapy treatment, triple negative cancer across multiple time stage. So here we just show pre post-treatment cells. on the tree, growing a pre, red or post-treatment cells. And we're asking whether we actually place the pre-treatment cell, actually going back, pre-treatment cells near the roots and post-treatment cell actually far away. Yes, our algorithm are able to do that, but standard algorithm were not able to do that. So we're actually truly infer the timing of the cells. Of course, we also observe a different kind of acceleration of sort of a genome instability. Once the cell acquires certain events, it seems to be copy number alteration process seems to be accelerated to some extent. This could be the transient instability Nick was talking about, but this definitely deserves a little bit more dive into because the model itself can have some bias too. So we applied this cohort of 22 BC from previous study, and we are able to identify the recurring events which are presumably functional. And we were able to record more of those events in standard algorithm, including genes which actually are associated with patient survival in the TCGA and the meta database. And this is also did by literature search over functional breast cancer genes, and also further validated by the knockout, CRISPR knockout data in the DepMap project on the breast cancer cell lines. So we're truly detecting functional genes. And one important thing to highlight is the parallel evolution, which can be a statistical model under our paradigm, because we're now looking for same genes which is recurrently altered in the same tree. And maybe it's in multiple sample. So this can be statistically rigorously characterized as a statistical test. So you can get a p-value out of this. So one thing come out is out of the 22 BC, we found 7 recurrently altered genes. So this seems to be higher than a point mutation in some other cancer types, suggesting copy number alteration is likely a better way to evolution occur. So we can also actually apply this to, okay, we can also occur this on the RNA, single-cell RNA sequencing data set to derive tree by apply algorithm like InverSynV, to convert RNA expression to copy number profile and then apply our approach. And we, analyze, for example, multiple myeloma, B-cell malignancy here. But we can also compare with the standard trajectory inference, since it's RNA-seq data, and to compare whether we better infer the time, for example, primary versus relapse, 2 different time stage. And yes, we're able to actually do better and infer in time than the just trajectory analysis. This is funny because we're looking at the same type of RNA data, but we're actually doing better in terms of time inference, representing the redundancy, the dependency of RNA expression actually have more information than the independent gene expression measurements. Again, we're able to validate this functionally by comparing the functional data to some extent, which is kind of interesting. Just a brief summary, we actually focused on many different areas of single-cell data analysis, but in this particular context, we looked at single-cell copy number evolution modeling. How do we computationally and statistically dissect those? This is going to be a long way to go. I think a lot of insight just coming out of Nick's study, so we really need to think further, how do we model the evolution of those copy number alteration? Quick acknowledgement of Fan Wang for doing much of the work, and it's a large collaboration with many people from Nick's lab and also collaborator at WashU. This study is funded by Chan Zuckerberg Initiative, separate, and preprint and source code are available online. I guess that's it. Thank you for your attention.
All right, thank you, Ken, for such a wonderful talk. If anybody has any questions, please post them into the chat window. If not, I'll maybe just start with one question first. Ken, so I'm wondering a little bit about MEDIC and how well it handles the issue that you brought up about, chromosome breakpoints are not just segments. Chromosome events can be large and overlapping and complex. And so how well do you think this method is able to handle, more complex copy number events? Even if we just have the read density data, there can be multiple breakpoints spanning across a copy number event before it kind of goes back to a ground state copy number. So how well is this handled, I guess, within MEDIC?
Yeah, I think it's definitely high some improvement over standard maximum parsimony approach where you basically have to segment the breakpoint for them to become independent. So you can code this into the matrix, integer matrix. So this algorithm does not actually require you to do that. You can actually segment the genome in a fixed interval. When the algorithm computes the estimate distance, this actual distance-based method is trying to look at how much of the interval is overlapping between two cells. So it tries to actually identify the biggest overlapping interval between the cells. So I guess, so it has a different kind of a genetic distance measurements than standard way. Of course, I think it's still vulnerable. The issue you mentioned is actually very complicated. It's still vulnerable. We're trying to just model this as a one copy gain and loss events over a longest possible interval, which is, I guess it's a very rough assumption for the complex events with lots of waypoints. It's actually pretty challenging to model, which is something deserve further thinking.
Okay, great. Thank you very much. Let's see. Any other questions for Ken? I don't see any posted to stream, so I think we can, if you have any questions for Ken, you can just post them in that window and Ken can kind of respond through using Brella. All right, well, thanks, Ken. That was a really wonderful talk. Oh, wait, let's see, one more question. This comes from Runman. The question is the tree branch length weighted? I guess he's talking about the MEDIC method.
Yeah, it's
or not MEDIC, sorry, your method.
Yeah. So the tree branchness right now is just the number of copy number iteration that is differ between two states. So it's an integer value. There's no weighting at this in the current implementation. Thanks for the question. By the way, before you sign off, thank you for the organization. I did not acknowledge and everybody for putting this conference together. So nice.
Okay, great. Well, thanks, Ken. Thanks so much. All right, so next we're going to move on to our vendor talks, and I'm going to hand it over now to Xiangwing Zhao, who's going to present the speakers that we have for the 10-minute talks that will be following from here. So thank you very much. I think we had a wonderful morning session today.
Okay, hello everyone. I hope you can hear me. So this is a vendor information session. We have 3 speakers from 3 companies. Each will have 5 minutes talk plus question answering. So the first speaker is Sean Piper from PinX Genomics. I hope you are ready, Sean.
Yes, I am.
Okay, very good.
Let's see.
So I will remind you about 5-6 minutes later.
Perfect. Thanks.
Mm-hmm.
Alrighty. Okay, hopefully y'all can see my screen. Hello, so as Dr. Zhao mentioned, I'm Sean. I'm the sales executive located here in Houston for 10x Genomics. And I'm really excited to give you just a brief overview of the 10x product portfolio and some of the updates that we've made this year. I don't have much time to get into really any depth on these products, but I will be available to answer your questions in the virtual booth during the workshops. I think those are at 12:50 and 2:50. So as you know, we really only understand a tiny fraction of the full complexity of biology, and this is the key to advancing human health and accelerating this understanding. So understanding these complexities at high resolution will be the key to genomic discoveries and ultimately accelerate the understanding of disease and therapeutic studies. And here at 10x, we've set out to drive this acceleration through our ever-expanding single cell and spatial applications portfolios. So we've seen a number of breakthrough discoveries across several different research areas over the past years where 10x has really been instrumental, from immunology and cancer research to research in developmental biology and neuroscience. Our customers have helped get one step closer to understanding the complexities behind these diseases such as cancer and Alzheimer's. Our solutions have really helped uncover some of the most fundamental processes that occur in human biology, including early stages of development, mechanistic insights into cancer resistance during treatment, and key pathways involved in neuropathogenesis. So the key to our technologies and platforms is the ability to take and interrogate biological samples at high resolution on the molecular level. Traditional platforms such as bulk RNA sequencing or limited marker stainings of tissue cross-sections often mask the underlying heterogeneity and miss critical information that is hidden behind average or incomplete measurements. Our Chromium platform is fundamental in dissecting tissues such as cancer biopsies at single cell resolution to uncover transcriptional and chromatin states that are unique to distinct cellular subpopulations. And while the Chromium platform can help answer the questions of what is expressed in a particular tissue, the Visium platform is the platform that addresses where and can spatially resolve unique expression signatures in heterogeneous tissues. So being able to resolve complex tissues and diseases on both the single cell and spatial level will provide us with more new insights into specific pathways, tissue microenvironments, and cellular networks. So over the past 5 years, we've introduced a suite of different applications, including single cell gene expression, single cell immune profiling, copy number variation, ATAC. And so kind of starting in 2019 is where I'll pick up. That's when we introduced our Visium platform, which was our first spatial gene expression solution. And then 2020 has been much more busy. We've launched our automation platform called the Chromium Connect. We've launched a targeted gene expression product that's compatible with both our 3' and 5' immune profiling solutions, immunofluorescence compatibility with Visium, and then also most recently our single cell multi-ohm ATAC plus gene expression kit. I guess also I should probably mention is that you might have heard this week about our 2 new acquisitions of both Cartana and ReadCore, and that's definitely going to expand our future spatial portfolios with in situ sequencing. So just a quick overview of the Chromium. This is just a fully integrated automated system that enables input of single cell or nuclei suspensions. It performs all the necessary steps to generate the sequencing ready library with minimal hands-on time. So it's just a complete walk-away solution that uses validated protocols and chemistries, and it'll produce consistent gene expression libraries. Right now, it's currently compatible with our 3' version 3 chemistries, but in early 2021, we're going to be enabling our 5' immune profiling kits just via our version 2 chemistry. So next, I'm going to briefly talk about multi-omic profiling, especially at single cell resolution. So last year, Nature named single cell multimodal omics the method of the year. And here at 10x, we're building these multi-omic tools to enable you to do just that. So many of our products already enable multi-omic profiling, such as the linking of transcriptome measurements to protein measurements, for example, feature barcoding or immune receptor profiling, among other data types. However, last month, we launched the first ever kit to profile both chromatin availability and gene expression from the same cell. So I can't really go through all of this kit, but long story short here, you can show direct linkage from your chromatin states to gene expression, as well as you can maximize precious samples by obtaining multiple readouts from the same cell. So I'm quickly going to go over a little bit of the Visium updates, but I just want to recap the workflow. So unlike single cell, this starts with a tissue section that's going to be stained with an H&E and then imaged. The reagents are then going to barcode and capture the transcripts and then create a gene expression library. And then finally, our analysis pipelines perform the downstream analysis and data visualization. So this past June, we launched Visium compatibility with immunofluorescence, which now enables multi-omic analysis with Visium. So you're pairing protein information with your gene expression information. So as a quick example, here where we stained mouse brain sections for the protein nu-in, and you can visualize these on the image here. And so if you carry the same tissue section forward through the Visium gene expression workflow, we can use our loop browser to visualize the mRNA expression. So here we're visualizing the gene RBFOX3, which actually encodes the nu-in protein. And so in this image, the darkest green intensity spots have the highest levels of this RBFOX gene expression. And then over on the right, you can actually see how we can combine the immunofluorescence data with the gene expression data, either from the single gene or from the whole transcriptome level. So this demonstrates the overall capability of Visium to combine your high resolution microscopy images with complete whole transcriptome sequencing readouts. Lastly, I can't mention Visium without without saying that right now it's compatible with fresh frozen samples only, but early next year, we're going to be launching separate FFPE compatible Visium kit. So just as a final thought, combining both single cell and spatial gene expression analysis allows you to address the questions of what, where, and how transcripts, proteins, and cellular interactions are influencing tissue biology and disease. This slide depicts a tangible analogy of how sequencing-based gene expression technologies can work together to better resolve our understanding of tissue function. Generally speaking, bulk RNA sequencing can give you a great heap of data, but without much insight.
Sean, sorry, you may have only 1 minute remaining.
Perfect, finishing up.
Okay.
The commercialization of single-cell RNA sequencing techniques such as Chromium single-cell gene expression products enable a better understanding of distinct cell types with their related profiles, but as dissociated populations. More recently, spatial transcriptomics techniques have helped piece together additional insights about cell types and expression profiles. And now many researchers are demonstrating the ways in which insights from single cell and spatial technologies build on each other to construct a more comprehensive and in-depth understanding of tissue function. So just to finish up, here's a list of our compatible partners for sample prep, feature barcoding, which such as the antibodies and the CRISPR guide RNAs, and our sequencing partners as well. And so, like I said earlier, if you have any other questions, you can visit our booth and during the workshops, and I'll be there to help you in the chat. So thank you, and thanks everybody for organizing this. This has been great.
Okay, thanks, Sean. That's really exciting. Because of time, we are right now behind the schedule, so we may move to the next speaker. And obviously, you can look at the questions from chat and reply them, Sean. So our next speaker is Steve Hoffman from Illumina, talking about the single cell technology from Illumina.
Good morning. It's great to talk to my neighbors here in the Gulf Coast about Illumina sequencing and how it fits together with all of these amazing single cell techniques that y'all are sharing, that you're using to understand biology. What I want to make sure I address is that Illumina NGS really provides a single NGS readout that can address a range of different applications, whether you're just QCing a library before you send it off to a service provider or core lab. If you're looking at a small panel, whether it's DNA or RNA, or doing protein or multi-omic, and even that spatial that Sean was talking about just a few moments ago. So, as a specialist at Illumina, the thing I get asked about most often is how much sequencing do I think you need for all of the different approaches to single cell genomics. And so here is, while it's not a have all or end all be all, this does really get you to the point of where to start the conversation for how many sequencing reads you need per cell or per sample for some of these techniques. It varies from tissue to tissue and cell to cell because of biology and the variation in terms of the number of genes or the number of proteins that are present in cells. But the other thing that I think is real important is just take this opportunity to share with each of you what's new at Illumina and how that fits into the single cell landscape. And if you are someone who has samples that come in, kind of one or two at a time, where you want to maybe run a handful of samples and get the data back quickly, the NextSeq 2000 is an update to the NextSeq 500 that really, first of all, it provides a bump in sequencing power with a reduced cost compared to that other benchtop platform. And it allows you to address what I think of as a standard single experiment very quickly with an instrument. It enables something on the order of a good match really for a whole 10x Chromium chip worth of samples can run on one of these P3 flow cells and generate, right about 1 billion sequencing reads. The other thing that I want to make sure that I mention is we've recently rolled out an update to the NovaSeq, which is a really high throughput platform. But with the new version 1.5 chemistry, if you compare the cost of doing really large single cell sequencing studies with the previous version 1.0 chemistry, you can really power up your single cell experiments for the same investment. If you're thinking about doing single cell RNA-seq rather than being able to afford 3,000 cells, you can add more conditions, more time points to your experiment, and get roughly 2 times the number of cells that you could have done with the previous chemistry. Or if you're doing a technique and you really are interested in those rare expression genes, now you can afford to bump the depth per cell that you sequence at up significantly using version 1.5 chemistry rather than the old version 1.0 chemistry. All of this together really just helps make the types of experiments that I think researchers are most excited about, those that include biological replicates and multiple time points, much more amenable. And the other thing that's really, I think, exciting about the version 1.5 chemistry is we've actually added a couple of more cycles worth of chemistry to these kits so that it can better enable UMIs and barcodes, including dual barcodes, so that you can really take advantage of all of these exciting ways to label individual cells or individual molecular reactions and not have to give up read length on your insert to be able to address those. So there's a lot of great features that will help you speed things up with the NextSeq, help you get more done in your own lab with the Dragon 3.7 update. We're going to have a universal demultiplexing built into the RNA-seq pipeline, and really happy to have you come by our Illumina booth and chat with your local sales team. Is going to be available there, and you can always drop me an email or chat me up in the box here as well. That's everything I want to talk about today. Thank you.
Okay, thanks, Steve. Let's see if there's any questions. I think there's no question there from my chat board. So, but obviously we can look at it later if some people raise the questions. This is very, very important technology from both single cell and the Illumina sequencing. So thank you again. And we move to the next speaker. So our next speaker is Jenny Roller from Protein Sample Company.
Hi, this is Jenny Rohr, and I am going to share my screen here. All right. Thank you. I hope you can hear me. And I will go ahead and start. Yes, my name is Jenny Rohr, and I'm with Protein Simple, which is a biotechny company. And I'm here with Dr. Tuan Dang. And we're here to talk to you a little bit about our single cell Western system down here at the bottom, Milo, and our simple Western system. So as I said, we are part of oh goodness, went a little bit too fast there. So, as I said, we are part of Biotechny, and with that, we have the single cell Western system, which is our Milo, our Western-based and protein detection and heterogeneity information, which really looks at 1,000 single cells per run and between 4 and 12 proteins per cell with a 4 to 6 hour workflow and 3 to 4 hours of that actually being hands-free, and uses your Western validated antibodies, just your typical Western conventional antibodies. So within the complete solution of Milo, you have your instruments, your single cell Western kits, your software, and just your typical antibodies. Milo is very fast and simple to use. You just simply load your cells onto the chip. You run your chip for 5 minutes, and you probe with your antibody cocktail of your primary and secondary. Then you read that chip with the microarray scanner to give you a TIFF image, and you analyze that in your Scout software, and that gives you many ways to look at your data. Within the chip here, this is what the chip looks like, and you have your single cells in suspension, and they're either trypsinized or adherent cells of dissociated tissue. And what you see right here is little micro wells that those cells go into. And it traps them via gravity using Poisson distribution, the key variables being concentration and settling time. And then you run it in Milo, where it will do a 1-minute SDS page size separation after it has lysed those cells open. It immobilizes the proteins into that chip using UV and making it very, very stable. Then you will start the incubation process there on your desktop with your standard Western validated antibodies. And then you use that image fluorescence with your microarray scanner. This is just a typical 2-color microarray scanner, and we're giving you 1/16 of that. And so within that, we give you even better view of that there were 6, oops, let me go back, sorry. There were 6 wells here at the top, and 4 of them were occupied by cells, and there was that 1-minute electrophoresis done so that it captured 3 actually targets within that sample right there. And that also can look at isoforms that way because you are getting size separation. You can also measure proteins that are hard to detect by FACS, by flow, by looking at isoforms and phosphorylated proteins, transcription factors, and intracellular proteins. You can also look at, obviously, get your protein validation from your single cell RNA-seq data. So what you do is you simply get your sample and you divide it. You can take some of that sample and use it, obviously, for your single cell capture and RNA-seq while you're getting your RNA and gene expression. During that time, you would just run a little bit of that sample in the single cell Western, and then you can hold it. Because of that UV cross-linking step, it is very, very stable, as I said, for about 9 months. And actually, you can run that same chip, strip and reprobe it at least 9 times. So that's great. You can look at a lot of things with that same sample. And then once you get your target expression, you probe it with those those targets that you have found through your RNA-seq to get your protein expression. And as I said before, with the FACS sorted populations, then you can get your cells, put them through to get your surface marker sorting, collected your subpopulation, and run them in the single cell Western to measure challenging intracellular proteins such as transcription factors and phosphoproteins on sorted cell subpopulations, and characterize highly enriched subpopulations with too few cells to analyze in flow. So this was also shown by a researcher here who, he had how many cells can express in one isoform or other or both, and was able to find that the single cell resolution enabled him to measure differential expression of each protein isoform and measure new classes of proteins in the single cell level, enabling new discoveries and insights. So, so they were able to really find the role of those 2 isoforms in their cancer models to gain new measurement and insight into the role of these isoforms, how they play in disease progression. So What can Milo do for you? It can measure efficient and low efficiency gene editing systems, simultaneously detect surface and intracellular proteins, analyze enriched cell subpopulations after fact sorting, validate single cell RNA data at the protein level, measure methylated histones in single cells, resolve protein isoforms, reveal hidden subpopulations, and study intracellular signaling. So it can do a lot. Really, you're looking at, think about just a typical Western, but on this single cell level. So it gives you so much more information, again, about heterogeneity and different subpopulations within your sample. So it really is opening up the world to answer so many new questions. So we have this, there's many, many new publications coming out daily on this because it is such a new technology that really just opens up the world to protein level information for single cells. So I also want to talk to you real quick about your simple Westerns, and that's a totally different platform if I just have a couple minutes. And really, if your Westerns typically look like this, we have the problem solvers, and you've probably seen them around. We have a lot of them here in Houston and all over Texas and all over the Gulf Coast. And you have this simple Western platform, which is your Jess, Wes, and all of the others. And really, this is an automated capillary-based electrophoresis system. And the reason for this, I thought you might want to know about it, is for two reasons. One is speed, because you can do these in at least 3 hours. It's a fully automated, start to finish Western in just conventional Western, automated, Western, looking at your average population. In 3 hours, you can do 12 to 24 samples, but it is very, very sensitive. It's capillary electrophoresis, and it is up to 100 times more sensitive than your typical Western, using very, very small amounts of protein. So if you have those really special and precious samples that you don't have many cells for, then this is the way to go as well. If you're still looking at that pooled average population, a typical Western, not just the single cell, but your typical pooled averages, then that's what this is for, because you're using about 0.2 micrograms per microliter, and you're only using 3 microliters. You can go even lower than that if you have a really good antibody. So that's the other one I wanted to talk to you about. And just real quick.
Yeah, okay, maybe 1 minute.
I was going to say, I had a feeling we were about done with that. So, so let me just go on. So because of the automated, we have reproducibility across multiple users and across multiple runs. So that's how your data look. Tuan and I, Dr. Tuan Dang and I, will be in the vendor workshop. We're happy to answer any questions you might have about either the single cell Western system Milo or the simple Western system Jess, Wes, or the larger systems. So thank you so much for your time. If you have any questions, let me know.
Okay, thank you, Jenny. Actually, I could not see the comments, questions from Bela because I'm logging from another system. So maybe a short question. Yeah, if someone, I see Nick has a question to Illumina. So maybe you can ask after my first question. So Jenny, I have one short question for you. The Milo looks like very, very exciting. So which kind of cost basis can you tell us a little bit about, how much to run?
Are you talking about for the chips themselves?
Yeah, more or less like if I want to run at a single level protein, Western, how much it is.
Oh, sure. So, so just for the chips themselves, you're looking a little over $100 per chip, and you can, and that gives you, that's all of the stuff that you will need, the wash buffers, the solutions, everything that you would need, except for your antibodies, and then. So, but you can strip and reprobe those, as I said, 9 times. We say 9, the NIH goes up to 12 times. So, and you can hold those chips just literally in a drawer for up to 9 months. So you can go back to that same population.
Yeah, Jenny, actually, there's another question for you. So to save time, what is the detection sensitivity for Milo and also amount of proteins in a cell?
Yeah, so that's a great question. So your detection sensitivity is, it really, I mean, if you have a very, very small cell with a very small amount of protein, then it will be a little harder. I have seen that, but where literally the cell size was about, I believe, 2 to 3 microns, maybe 2 microns, and it had a very small amount of protein in it. That one we had problems with. But on your normal size, most cells are around the 9 micron to 12 micron range. We go up to at least 25 microns in our different sizes of micro wells and chips. Then you can, you know, it's just, it's each cell obviously has to have some protein in it, and some people are even looking at nuclei as well, protein and nuclei in different parts of the cell, intracellular, extracellular. So it just we have been able to see almost every type of cell with this system.
Okay.
So
yeah, thank you, Jeannie. Nick, if you is you have a question to Steve Hoffman, Illumina, otherwise we can finish here. You can ask one more question. I guess probably, yeah. So for all the speakers, maybe you can go to Bella postboard to answer the questions. So it's just right time, 11:40. So we finish the vendor information session here, and we move to the next session 2. Thank you all for all the speakers.
thank you.
My pleasure. See you later.
Okay, so sorry that we actually are running a little bit late, so we don't have time for a break. We're just one session away from lunch, so thanks for staying with us. I'm Hongyu Miao, Associate Professor of Biostatistics and Data Science from University of Texas Health Science Center at Houston. So the potential of single-cell omics is amazing to me, and I'm lucky to have some experience with this technology where collaborations with our local investigators, including Dr. Ray Chen. So the focus of session 2 is the application of single-cell omics in immunology, metabolism, and cell differentiation. And I'm very excited to have the opportunity to introduce you the speakers. Our first speaker of session 2 is Dr. Gunaranti. So she is the professor of the Department of Biology and Biochemistry at the University of Houston. She got a PhD in genetics from Cornell University. After 3 years of postdoc in Chicago, she came to Houston and stayed with us. Dr. Gunaranti primary interests include non-coding genome and the regulation network. She has many publications on prestigious journals, including Nature, Nature Communications, Nature Genetics, PNAS, and Cancer Cell. Single-cell technology obviously became one of her new favorites, and she will give a talk today on response signatures of NK cells and CD4, CD8 cells. So let's welcome Dr. Gunaranti.
Thank you, Hongyu. First of all, I'd like to thank all the organizers of this very exciting symposium and for the invitation to give a talk. So as director of the US Sequencing Corps, I have had the good fortune of being involved in a number of collaborations involving single-cell sequencing. But today I chose 2 collaborations to present, which I found the most exciting. And the first one is a collaboration with Emily Mays, who, if you all recall, was originally at Texas Children's and Baylor, and now at Columbia Medical School, on the discovery of 3 new natural killer cell populations using single-cell transcriptomics. And this will be followed by findings from a collaboration I had with Honglu Wu at NASA, using single-cell transcriptomics to delineate a T-cell specific radiation response signature. So now a little bit on natural killer cells, which I had to also educate myself, being with my main focus being cancer. These are cytotoxic lymphocytes that have been implicated in a wide variety of diseases, including viral, microbial pathogen defense, autoimmune disorders, reproductive complications, and a big involvement in detecting and killing cancer cells and critical components of transplantation, cancer immunotherapy, and so on. And they are really key regulators of the innate and adaptive immunity. So basically, they are a type of lymphocytes, just like T and B cells. By contrast to T cells, for instance, they do not need priming prior to activation. So often they are the first line of defense, right? They secrete cytokines such as interferon gamma, TNF alpha, and they act to then enhance immune response by signaling to other immune cells like macrophages and dendritic cells. So there are two functions. First is a direct function, the first line of defense, innate immune response to an infected cell. They start secreting perforins, granzyme B, and cause a cytotoxic response to cause apoptosis, necrosis, and so on. But in addition to that, natural killer cells that are activated by cytokines then function to secrete more and more cytokines to then send signals to trigger a widespread adaptive immune response to B cells and T cells. So they become very important components of our immune system. So if you look at the typical immunotypes as defined by CD56 receptor, there's the CD66 bright, which is the cytokine producing cells, the CD56 dim, the cytolytic cells, and then the CD56 negatives, which are impaired NK cells. These are the other various receptors that each one carries, including killer immunoglobulin-like receptors that interact with MHC class I molecules. So if you look at the three types, the CD56 brights have secrete a lot of cytokines, little to medium chemokines, have little low cytotoxicity, but highly proliferative. The CD56 DIMs have high cytokines, they're very active, high chemokines, highly cytotoxic, less proliferative. And then the impaired ones have little or no cytokine secretion, but still has high levels of chemokine secretion, and then very little cytotoxicity. So basically, they are generated from the hematopoietic stem cell, undergoes several stages of maturation, and then basically sits in lymphoid tissue until there's a signal from an antigen, at which point they become, in the frontline defense, one of the largest circulating populations, right? And so the life cycle is these NK cells go from being naive cells, activated by antigens, to cytotoxic cells, and then they start secreting interferon gamma cytokines to signal to other cells and engage other cells and become regulatory NK cells and become a very important part of adaptive memory, so that the next time that the cell is presented with the same antigen, they are able to launch a very strong attack which is much stronger than the initial one. So with Emily, what we did was they isolated blood NK cells from 2 healthy CMV negative donors. They were age matched, they're 60-year-olds, and it was a male and a female. They were treated, they looked at treated with IL-2 for 4 hours. These are called stimulated, versus untreated controls from the same donors, right? And about a little under 8,500 individual cells were sequenced with these read numbers, and unsupervised clustering then revealed about 10 subsets of these, right? The NK cells were isolated from PBMCs by negative magnetic bead selection. These are the flow cytometry plots. These are the total percentage of CD107A1 cells in donor A and donor B. And then this is the Tisney plot with the 10 clusters. This is a heat map with the clusters with the genes, and almost off the bat, we were able to exclude clusters 5, 7, and 9. 9 looked very much like they were antigen presenting cells. Cluster 7, the gene expression signature indicated that they were likely T cells or NK T cells. And cluster 5 expressed a disproportionate number of heat shock proteins and DNA repair genes, which we took to mean that this was a technical, had an issue with the isolation process. So the remaining clusters, so before we performed single cell sequencing, what was known is that human peripheral blood NK cells are classified into 2 developmentally related but functionally distinct subsets, distinguished by the CD56 dim versus bright status. The dim CD56 dim are the cytolytic NK cells. The brights are the adaptive memory NK cells. And then there's an intermediate phenotype carrying these markers. And then, of course, the impaired cells. And now I will take you through each of the clusters that we found. We found cluster 2 to relate to a CD56 dim 16 plus 57 plus activated NK cells like phenotype. Here you're looking at the percentage of individual NK cells expressing 1, 2, 3, or 4 different killer immunoglobulin-like receptors. On this side, you are seeing the dot plot of where the number of the size of the dot represents the percentage of cells expressing these markers, and the color of the dot represents an average scale expression value, with dark colors being high and light colors being low. And this is the signature for cluster 2. Cluster 3 is where we found a previously unknown, unanticipated subpopulation of that looked like a type 1 interferon responding subpopulation. And this is the gene expression signature for that. As you can see, high levels of CCL5, and then the relative levels of these in relation to the other clusters. And as you can see, some, good number, proportion of cells were expressing this cluster. Cluster 4 looked like it represented the CD56 bright NK cells responding potently to IL-2. As you can see, very high expression of these genes. The proportion of cells were lower, but whatever cells that were expressing this was expressing the signature at high enrichment. Cluster 6 then, these were NK cells displaying a cytokine-induced memory-like phenotype, and that's cluster 6. And this is the signature for cluster 6. And then, as you can see, some large proportion of cells were also expressing that. And then we found another previously unknown cluster, a new subpopulation exhibiting a great loss of ribosomal gene expression, right? And interestingly, high expression of MALAT1, which is a long non-coding RNA. So this is a summary of what was found on the 7 clusters and the Tisney plot. About 1/3 of the population represents the CD56 dim cells. About 25% were active CD56 dims. About 1/5, 20% represented the adaptive NK cells with a signature high expression of these genes. And then there were small proportions of other cells, including the ones we excluded that were not NKs, and the novel subpopulations. So after single-cell RNA-seq, these were known to exist previously. These represented the new subpopulations that we discovered. The first one is a CD56 negative type 1 interferon responding NK cells, closely related to the CD56 dim cells, defined by its response to type 1 interferons. If you notice that they carry receptors, And they exhibit a cytokine-induced memory-like phenotype. They have increased granzyme B expression in response to IL-2, and then specialized communication with dendritic and T cells through the secretion of these factors. The other novel subset that we found is CD57 plus NK cells with significantly reduced ribosomal expression, showed downregulation of oxidative phosphorylation and a high level of immediate early response genes indicative of cellular activation. And then there was this curious population that looked like a mix. It was CD56 dim, 16 plus, 57 minus NK cells, had greater amounts of chemokines and a higher proportion of KIRs, and a higher level of 4-1BB. Responses to IL-2 were very similar in both. They also then took a CMV positive donor, which was a younger female, about 40 years, and also compared the single cell sequencing data with the CMV negative donors, and found that there were altered proportion of NK cells and gene regulation in each subset, with a significant increase in adaptive NK cells. So together, the data, I found very exciting. It established an unexpected diversity in blood NK cells and provides a new framework for analyzing NK cell response in health and disease. Especially, we are excited about this type 1 interferon responding NK cells because COVID-19, as it enters the cell, one of the first things that the spike protein does is, and some of the non-structural proteins from the genome, is to block the interferon reaction. And the question is how to harness these NK cells to counter infection. This work was published earlier this year in April in Blood Advances. In the next segment, I want to share with you what we did with NASA, with Honglu Wu. And that was, they were interested in looking for a cell-specific radiation response signature from human lymphocyte subpopulation, right? So as you know, detrimental health consequences from exposure to radiation from long duration flights are a major concern at NASA. And we all know that the cellular responses to radiation where only high energy protons and other particles traverse fractions of cells are expected to be heterogeneous. So assessing DNA damage and damage response at a single cell resolution becomes critically important. So when astronauts are in space over long periods, they run the risk of exposure to radiation, which has been documented to cause chromosomal aberration, cell death. A question is, can we use single cell transcriptomics to discover gene expression signatures for radiation risk, with the lofty goal of one day developing a biomarker for remote sensing precancerous changes through a blood test, right? So that was the goal that we all set out to. Honglu and Maria Moreno Villanueva took primary human T cells that were isolated from PBMCs, and then and looked at 3 hours after ex vivo exposure to gamma rays, right? So they are simulating space flight. Single cell RNA was performed on 700 irradiated and compared with 700 non-irradiated control cells with about 50,000 reads per cell. And the PCA clustering found 3 different subtypes, CD4 plus naive CD8 NK cells. And These are the relative sizes of CD16 intensity gated CD45s. These are the knee plots. The average size was about 500 base pairs. And this is the t-SNE plot showing the 3 groups. When we looked at the gene expression signatures, what really popped out was that genes involved in P53 signaling pathway was highly activated upon radiation exposure, and the STAT1 IRF1 signaling pathway was also activated, right? So, but the conclusion was that the 2 primary pathways in response to radiation exposure in T cells is P53 and STAT1 IRF1. And these are some of the qPCR validations from the same groups. And pathway analysis, this is pathway analysis showing P53 activation as the primary response to radiation exposure in human T cells. And there's also activation of STAT1 IRF1. The TP53 responsive genes and genes that are involved in the interferon gamma pathway are activated. These 3 genes, BBC3, PCNA, and TP53 related genes that have been known to respond to radiation show increased activation. Most of the TP53 genes were upregulated in all 3 groups of T cells. But IRF1, STAT1, BATF were upregulated in the CD4 and 9 groups, but remain unchanged in the CD8 and NK group, which suggested that the interferon gamma pathway does not respond to radiation in the CD8 plus and NK cells. This work was published about, in April of last year. And once again, this is a collaboration with Honglu Wu and Maria Moreno Villanueva. I thought it was really exciting that we have the first hints of a radiation response signature, which, like I alluded to, the ultimate goal is to find biomarkers, blood biomarkers to predict precancerous changes in that follows radiation exposure during flight. I'd like to end by acknowledging the various collaborators, Emily Mays and Sung-Mei So at Columbia, and Honglu Wu and Maria Moreno Villanueva at NASA. The key lead in our lab for both these projects was Brandon Mistretta. He's just a graduate student, just finishing up his PhD. He's also the lead scientist at the US Sequencing Co. Also Ying Hong Pan, who's now at the UPMC Genome Center, and Sujas Chatterjee, who went on to join QIAGEN. And of course, I would like to acknowledge the funding agencies, NIAID, and with a grant to Emily and me, and NASA to Honglu Wu. Thank you. Take any questions.
Thank you. Any questions? Because right now, I haven't seen any questions in the chat channel, but I do have one for the speaker. So I think this is very interesting work, and because I have been working on immunology questions for a long time. So I'm wondering, what are the unbeatable benefits you get by using single-cell technology to investigate this kind of immune cells?
I think it's amazing. I mean, we did not expect, as you saw, the proportions of cells were smaller, right? That carried the new subpopulations was smaller, right? And when you do bulk RNA sequencing, you're going to lose that signal, right? And the amazing resolution one gets from single cell sequencing, was really evident to me from this study, as you all know. And along with the proportions, I think keynote speaker made a point that not only do you get gene expression signatures, associated with subpopulations, but you also can tell the proportions of cells that carry the signature, and the degree of enrichment, I think, is hugely powerful. And the ability to capture the rare cells, right? I mean, in the cancer field, we are struggling with finding the cancer stem cell, the rare cell populations that drive things. So I think like this project to me was very exciting to uncover, 3 new subpopulations.
Thank you. A question from the audience. If a cell subset were CD56, how do you distinguish from T cell?
So I have to confess I'm mostly a cancer person, and Emily Mays, my collaborator, was going to be on the call, but she had other, she couldn't engage. So I can, check with her and get back to you. But am I right that there are enough receptors on T cells versus CD56, the combination of receptors can tell the difference between NK and T. And I know there is a group that is, if you recall, we even eliminated here the NKT cell group. But I will be happy to get back to you after I talk to Emily on that.
Thank you. So now let's welcome our last speaker of this session. Dr. Han Ruan is a young scientist from UT Health Science Center at Houston. He received his bachelor's degree in biology and master's degree in ecology from Nanjing University. He worked at Novogen as a bioinformatician before he studied as PhD students at Fudan University, where he earned his PhD degree in bioinformatics. He now works at Dr. Len Han's lab at UT Health, where his research focuses on mining the multidimensional omics data for novel biomarkers and developing comprehensive data portals. Of course, he must have publications on Nature Communications to be qualified for this session, but that doesn't prevent him from publishing on Nature Metabolism, Nature Genetics, and Nature Biotechnology. So I will turn it over to Dr. Ruan for his talk on differentiation trajectory today. Welcome.
Okay. Oh, thank you for Dr. Miao for this kind introduction. So good afternoon. My name is Han Ruan, and I'm a postdoctoral fellow in Dr. Len Han's lab at UT Health. So let me share my screen first. So at first, I'd like to thank all organizers for hosting this single-cell omics symposium during this special time. And it's my great honor to do a remote oral presentation here today. So I'm going to talk about our lab recent single-cell application work on the subject of cardiac differentiation. So this is the outline of my talk today. So background for which inspire us to do the project and the design for our single-cell study, and a series of computational analysis of single-cell RNA-seq data from which we have some interesting findings. And at last, we took several steps forward for more sequencing and experiment for further validation and of the signals we dig out from the single-cell data. So first, a little bit background for what inspired us to do the project. So in virtual differentiation of human embryonic stem cells into cardiomyocytes is a very useful model system. So they have provided us the opportunity to better understanding the molecular mechanism underlying heart development, and even further to better understanding of the cardiovascular disease. So previous studies has been using the NGS technology like the RNA-seq and ChIP-seq to better understand the whole process, like this 2012 Cell paper, they have used the RNA-seq data to highlight molecular features at the transcriptome level. So by using the conventional clustering and differential expression analysis, some marker genes could be highlighted because they are highly expressed at a certain development stage. So inspired by this study and a bunch of other studies adopting this HSC to CM differential system, so why now we have the single cell technology, so we could do something further. So here is the experiment design for this study. So the protocol applied here is a very popular one. It includes 3 steps. First step, induction of methadone through activation of WNT signaling pathway between day 0 to day 2. And second step, between day 2 and day 5, so while suppression of the WNT signaling pathway using small molecular inhibitors, cells they are committing to cardiac precursors. And the third step, and cells are specifying into cardiac fate after day 5 upon extended culture in the Bayesian medium. So we harvest during this culture process, we harvest single cells at 6 time points, including day 0 to day 60. So this time point marks certain development stages, as the previous study would suggest. And total was 7,000 cells were harvested and sent up to Takara ICL8 single cell platform for library building and sequencing. So we end up with 6,800 of the high quality single cells for the following analysis, as shows in this t-SNE plot. So while we got the data, the first step is to comprehensively explore the gene expression pattern. So the expression pattern of the long differentiation stage-specific markers, they are correlated well with the time point or development stages. For instance, gene marks potency was strongly expressed at early stage, and gene marks for cardiomyocyte, they was highly expressed at later stage, and gene marks for early cardiac markers, they're such as ISO1 and MEF2C. So they're expressed around the day 5 and the day 9. So if we take a look at the clustering of the human transcription factors among all the single cells in this heat map, we could also spot the similar pattern. So they all expressed and well correlated with the previous knowledge we have known. So what we don't know, so it's a step forward to explore the overall expression pattern. So because t-SNE plot is not going to tell the relationship between the cell populations, we cannot tell how close these 2 cell populations are. So we try another approach called the manifold learning. So this approach, we learn, enable you to learn intrinsic tree-like structure of the data from all the time points. So we build the cell differential tree on the top differential expressed transcription factors. So we are choosing TF to build the tree as the more upstream in the context of the gene regulation. And we can observe the bifurcating point, so on day 5. So what does that bifurcation point mean? So it probably indicates a cell fate diverge or something happened on day 5 or earlier. So that's just assumption. We don't know if it's true, so we have some further analysis. So when we took a close look at the data by just taking the 4 time points around the bifurcation point, and this time we're trying to identify subpopulations as the most single cell studies would do. So we manually check the cell markers and do some enrichment of several marker groups. So we came up with several putative subpopulations of each time point. So for instance, we could spot a small population of cardiac progenitors on day 5. So because the t-SNE plot could not represent relationships between subpopulations, so it is not a trajectory. So again, we adopt a manifold learning approach and reconstruct the trajectory from day 2 to day 14 based on the top differential expressed genes among the time points. Then we map the subpopulation information of the cells detected earlier onto this trajectory. So we could spot a potential cell fate bifurcation point on the day 5. So we have highlighted these two populations, two day 5 populations, the androgen cells and the cardiac progenitor cells on this trajectory, as this seems to be on the two different paths of cell fate. So look at these two cell populations. So we have assumption. So it has been reported that in vivo, these androgen cells, they are supporting cardiac progenitors at a certain level. So is this kind of cell-cell crosstalk be observed in our data? So we manually compare the gene expression between these two populations. So from this volcano plot, we found that several highly expressed genes in the cardiac progenitor cells. We spot 2 TF highlighted in orange here, MAF2C and ETS1. MAF2C, which is myocyte enhancer factor 2C. This is a well-known factor that for mediating cardiac development. But ETS1 is not reported before as an important factor as MAF2C. So what's the role of the ETS1 here? That's what we're going to talk about later. Here, we have also found that 2 genes, KDR and FLT1. So they are encoding receptor VEGF2 and VEGF1 in the VEGF signal pathway. So under assumption that these androgen cells could be functioning as a supporting role to cardiac progenitors, and we have observed VEGF receptor genes. They are enriched in cardiac progenitors. So we take a look at the expression pattern of the paired ligate genes like the VEGF-A and PGF, and found that they are highly expressed in androgen cells. And if we visualize the entire VEGF signaling pathway between the day 5 cells using another tool, so in this figure here, the out cycle on the day 5 single, the out cycle is all day 5 single cells, and each line between the cells there's represents a significant possibility that the signals are being passed between cells. So we observe that androgen cells here, they are contributing a great portion to the cardiac progenitors in the context of VEGF signaling. So computationally, we observe that the crosstalk between the androgen cells and the cardiac progenitors could exist, and they are potentially regulating cardiac energy commitment. So after that, we examine some pathways like VEGF, MAPK, RAS using the gene set enrichment analysis in the cardiac progenitor cells. So because they're functionally known to induce MF2C and ETS1 expression, so they're all significantly enriched in the cardiac progenitor subpopulation. So that could partially explain why MF2C and ETS1 are highly expressed in the cardiac progenitors. So from all the computational analysis above, we could say that the crosstalk between the cell subpopulations and activation of their downstream signaling pathways, and they are responsible for the higher expression of these 2 TFs. So because ETS1 is there seldomly mentioned before in their role in cardiac differentiation. So we have to shift our focus onto this very TF. So what's the role of ETS1 in the cardiac energy commitment? Here is something we do. When plotting the expression pattern of ETS family and other well-characterized cardiac TFs in the subpopulation from the 4-time point, we plot the dynamics of their expression in the studio time space. We found that expression of ETS1, it displayed a unique pattern that is distinct from other cardiac TFs. even from the members from the ones in the ETS family. So let me zoom in on ETS1 so you can see that its expression elevated at end of day 5 and peak on day 9 and dropped again on day 14. So we want to further explore its regulatory rule as ETS1 is a TF. So we applied another computational method. There's specific design for single-cell RNA-seq data to predict the ETS1 regular activity. So the regular activity is a score to measure the putative downstream targets of certain TFs. So if we map the predicted regular activity AUC onto the cell differential trajectory, we could find out that ETS1, the regular activity, there remain low on day 2 and increase on day 5 and peak at day 9, and after which it dropped abruptly. So all of which this pattern resembles its expression pattern. So based on the previously computational predicted regulatory activity of ETS1, we went a step forward to access its genomic binding signals using the chipset technology. Here is an example of TNNT1, which is a very characterized cardiac structure gene. So gene tracks show here that ETS1 occupancy at the transcription start site of TNNT2 is really at its peak on day 9, so which is consistent with our computational prediction of ETS1 regular activity. So when examining the gene expression of ETS1 binding of the cardiac TF and those of cardiac structure genes, so we find highly correlated expression level with ETS1 binding of these two groups of genes. All these patterns suggest that there could be a direct role of ETS1 in activating expression of these genes. And we perform another validation of ETS1 lockdown at the cardiomyocyte stage. So this is real-time PCR to show that the expression of cardiac genes, they significantly went down when knocking down ETS1. So to conclude our study in one sentence, that is by computational analysis of single-cell RNA-seq data, specifically by reconstruction, the differential trajectory around the cardiac lineage commitment stage, and with some further validation, we found that one TF, ETS1, has important downstream factors that could be the result of cell-cell interaction to regulate the cardiac lineage commitment. So because of the relatively small size of the cardiac progenitor cells on day 5, this signal we present here could only be detected under higher resolution using the single-cell technology. So that's all for my talk today. So I'd like to thank Dr. Lin Han and the lab members and other collaborators who contribute to this project, and thank you all for your attention.
Thank you, Han. So any questions for Han? So actually, I think for this kind of differentiation trajectory research, it's always a big concern for the validation part, right?
Yeah, sure.
So can you actually explain a little bit more about the what kind of validations have you done to comprehensively to support your conclusion?
So the first validation is that we use the chipset data to show that ETS1. Sorry, I can't go to this slide. To show that actually this ETS1 is actually binding on these important cardiac structure genes. So this fit out with our prediction of the regular activity. Another one is that the knockdown of ETS1 at the cardiomyocyte stage shows that ETS1 is actually functioning as important transcription factor.
Yeah, that will be a very good validation. But how about the like the timing or the order of the trajectory?
Okay, let me go to this. This slide. So you mean the timing and order?
Yeah.
Okay, so because we have 6 time point data, but we only use the 4 time point data. So in order to give a very clear indication of how the trajectory looks like, because we are using the Monaco tool to reconstruction the trajectory. So this sometimes you tuning the parameters, the result will become different, but we can kind of have some trying of a different top differential expression gene to build the trajectory. We find out that that this kind of trajectory is kind of solid as we try different gene sets. So we think that this trajectory is kind of fit for our further analysis.
So they're validated based on knowledge or reasoning, not based on experimental results, right?
Yeah. So the trajectory is purely based on the computational, unsupervised computational prediction. But we have not a further validation of this trajectory. Okay.
Okay. Thank you.
Okay.
So
thank
other question? If not, I'm gonna let's thank Han again. Okay, that's our last speaker of session 2. I'm gonna turn it over to the poster session. Thank you, Han. Thank you all.
Okay, so welcome back everyone to the very last session of this great symposium today. And in this last session, we will have 3 speakers. Each one of them will have 25 minutes for the talk, as well as for questions and answers at the end. By the way, I am Luay Nakli from Rice University. So our first speaker for now is going to be Dr. Lucas Simon. Lucas finished his PhD degree at Baylor College of Medicine in 2016. He then followed that with a postdoc at the Helmholtz Research Center in Munich, where he worked mainly on method development for single-cell RNA. After that, he came back to Houston and he joined UT Health School of Biomedical Informatics as a research assistant professor. He is now group leader of computational biology at the Therapeutic Innovation Center at Baylor College of Medicine. His main focus is on understanding human disease, with particular emphasis on single-cell RNA sequencing data. And with that, take it away, Lucas.
Thank you. And I will assume that you can hear me and see my slides. If not, just feel free to interrupt me. Thank you for the kind introduction, and thanks to the organizers for giving me the opportunity to present. We've heard a bunch of fantastic talks about single-cell RNA-seq and other technologies. So I want to start my talk with a very basic question. I want to ask, what is a cell? And borrowing the definition of Stephen Quake from earlier today, he described it as a bag of RNA that can be computationally described as a 20,000 dimensional vector of gene expression values. And I think that's the right computational description of a single cell. But what's the representation of a cell? So to me, this is actually a representation of a cell. It's usually a dot in front of a lot of white space. But just like a GPS system is sort of useless with a single coordinate, it's really important to understand, what's the position of the cell with respect to cells of the same state, cells of the same cell type or class, and then across the entire tissue. So this is how we build these cellular maps, and really using these cellular maps is where we kind of infer biology from. So what's really important to give meaning to a cell is the coordinate system that underlies these cellular maps. And this is sort of what I like to focus my talk on today, this somewhat underappreciated white background in many scientific publications that we want to dive in deeper and really understand how fluid and dynamic this white space actually is. So I just want to quickly review something that came up in the other talks when you're comparing single cell to traditional bulk. From an analytical perspective, there are three key differences, which is that you have many more data points. The data is more complex, it's higher dimensional, and the data is also more sensitive. So you can capture cells in certain cell cycle states, but at the same time, it's also more sensitive to noise, such as, for example, dropout effects or others. So we're interested in machine learning, and for any sort of data scientist, these three attributes make single cell RNA sequencing data very exciting because it provides a great playground for applying more sophisticated analysis techniques than in traditional bulk RNA sequencing data. And we really started with the idea, okay, we have many more data points, but the data is more noisy. Can we somehow leverage using more data points to correct our expression data? And so the goal here was to denoise the data. And to do that, we use a technique sort of called imputation. And I think there's a nice analogy to explain this technique by discussing the video streaming service Netflix. So Netflix has a bunch of users in movies, and every user sort of ranks the movie. And then the algorithm will sort of predict for each user what movie they would like. And so first of all, this only works because there is big redundancy in the data. That means while there's 1 million users on Netflix, there's not 1 million unique movie tastes, but the intrinsic dimensionality is much, much lower. And that is sort of a key message. And it's somewhat ironic then that when we get all of these personalized AI recommendations that in all aspects of life, you know, and if we actually do feel like these are good recommendations, then it just means that we fit the stereotype of the algorithm really well. But of course, in our case, we don't look at users and movies, we look at cells and genes. And so more specifically, the concept that we're looking at is manifold learning. So we have this high dimensional gene expression matrix that consists of cells and genes and exists in this high ambient space. But the true underlying biology is much lower. And for example, if there was a differentiation or developmental progress, you could sort of illustrate or represent this progress as this black curve here. However, when you're actually making observations, so when you're sequencing the data, you observe data points that can be corrupted, and they don't lie exactly on top of the data manifold. They are sort of corrupted due to sequencing noise or other technical artifacts. However, if you do know the data manifold, then it's pretty easy to kind of correct your observations and move the data points onto the data manifold. And this is sort of the idea for denoising. So there are multiple techniques to do this denoising, and some posters actually at this symposium, they deal with this concept as well. What we used here is an autoencoder. An autoencoder is a neural network that actually learns itself. So the input is the same as the output. This sounds very trivial, you know, if you could just have the information flow through, but there's one little hook to it, which is that the bottleneck layer is lower dimensional, and it's usually restricted quite a lot. So in that sense, you know, the autoencoder has to learn an efficient compression to be able to reconstruct itself. And in our case, so that the input is our expression matrix, and we want to get our denoised output. And this approach has a few advantages. It sort of allows you to capture nonlinear relationships. It's highly scalable to many cells. And in our case, we implemented an adaptive noise model that works with count expression data. And if you're interested in sort of the mathematical explanation of it, you know, it's described in our manuscript, but I will try to give you a more intuitive explanation of what's going on. So the autoencoder is actually similar to our brain in the sense that we also don't remember everything or memorize everything. We try to compress information. And now it's already late afternoon, and our bottleneck layer in our brain is probably somewhat smaller dimensional than it was maybe in the morning when we had coffee and we were fresh. So now if you were an autoencoder and I task you with learning a data set, and I show you this data set, and then I ask you, okay, can you recite the numbers I showed you? You would have trouble, right? On the other hand, if I show you this data set, and I'd ask you now, can you recite the data set? You would do that fairly easily. And why? It's because you actually understood you didn't memorize every single entry in this matrix, but you quickly identify the data manifold, right? Which is that you have an increasing sequence of numbers in the diagonal, and the rest is zeros. So, if you have this data set and I ask you, can you predict the next data point without knowing the data manifold, it's going to be tough. But the real answer would have been 03761. And now for this data set, right, you know the data manifold, it's pretty straightforward to predict the next data entry. So having said this, now if I corrupt an entry or a deviation from the data manifold, it's much easier to identify it in the right data set compared to the left. And that is sort of an intuitive explanation of how the autoencoder works. So in our case, we applied it to simulated single cell data. So here we simulated 2 different cell types essentially, and you can see that they are nicely separated. When we add single cell specific noise on top of it, including dropout, this clustering structure is lost, right? However, when we denoise the data with our autoencoder, we can actually recover some of this clustering structure. And that is not the case if you use a standard autoencoder, which is based on a mean squared error. So that was the real application. We wanted to denoise the data, but we actually made a different insight into the data that we didn't think about in the beginning, but I think in retrospect was much more important. And so we asked, what's going on in the algorithm? How does it actually know how to denoise the data? And so the nice thing about neural networks is that you can actually visualize the activations of neurons in the network. And so in this case, we forced the autoencoder to do extreme compression and define the bottleneck layer to be 2-dimensional, because then we can actually just visualize the activations of these 2 neurons. So we applied the autoencoder with a bottleneck of 2 neurons to PBMC data, and this is what we got. So this is essentially the coordinate system that the algorithm or the autoencoder gave it to us. And now if I sort of overlay cell type information, you will see that it actually managed to come up with a coordinate system or representation of the cells that is meaningful in the sense that it separates different cell types. So that was a great insight from our first analysis. Then we asked, okay, what happens if instead of providing sort of the entire transcriptome space to the autoencoder, what if we restrict it to a small set of genes? And not just any random genes, but annotated gene sets. So what would happen in that case? We applied this to a data set that profiled interferon beta stimulated PBMCs. So there was this interferon beta stimulation. And now if we look at any sort of random gene set, in this case, this is a gene set of genes that are annotated as protein secretion, you will see that in this coordinate system or in this embedding, the cells sort of fall randomly with respect to the outcome. However, if you have a relevant gene set, in this case, interferon gamma response, you will actually see that the cells separate quite nicely. And so we developed a way to actually quantify this non-randomness, and we call it relevance score. And this relevance score now allows researchers to rank gene sets based on their relevance to a specific outcome or variable of interest. And so if you're interested, you can check out our GitHub. It's called Driver. But I also want to emphasize that these two plots that I'm showing here, they're showing the same exact cells. So the cells and the underlying 20,000 dimensional vector is exactly the same. But depending on what perspective you look at, you get 2 very different pictures and images. And I think this is really important to remember that the data manifold of single cell RNA sequencing can be very dynamic. So the same set of cells can look very different from different angles and different perspectives. And so as Ored mentioned, this Human Cell Atlas project, I don't want to repeat it, but I do want to point out that the whole concept of the Human Cell Atlas really embraces the community and the heterogeneity in the community, where everybody can contribute with their own sort of protocols, their own experiments, and so on. So these numbers might be a little bit outdated, but the point is that there are many, many different data sets coming from different technologies. How do you integrate this data? And so now if you look at a simple example, here we look at 4 different studies that all looked at pancreas cells. So it's the same tissue containing the same cell types, just profiled from 4 different groups using different technologies. And on the left side, you can see that the UMAP embedding here, a standard approach to visualize single-cell RNA sequencing data in reduced dimensions, shows that most of the cells, they cluster by study. And you can see on the right side that instead of one sort of cluster of alpha cells, they're colored purple, you see 4 different ones from 4 different studies. So this coordinate system of the UMAP doesn't really make sense, or it's not meaningful for the interpretation of cell types. And that's why we try to come up with a way to define a coordinate system that is free of batch effects. And so, yeah, the goal is, we have this single cell embedding and we want to generate an integrated embedding. And what we use as an approach is called triplet neural networks. And this is a neural network that sort of comes from imaging analysis. And instead of feeding a single data point at a time, you always feed a triplet of data points where you have an anchor and a positive, and those 2 are of the same type or class. In this case, this is an image of President Obama. And then as a negative, the 3rd data point in the triplet is a negative example. And this is actually how we also believe babies learn different concepts. They don't need to see 1,000 elephants to understand an elephant. Maybe they've seen some cats and dogs, and then they see that the elephant is different. And that difference is really what they remember, not every sort of attribute of the elephant. So we implemented this triplet neural network in a framework that we call Insight. And of course, in our case, when we're dealing with cells, we don't really have classes. So we don't have a label of this is President Obama, this is Macron. We actually have no labels. But what we do have is sort of existing tools to identify transcriptional similarity across different batches, such as KNN or mutual nearest neighbors. So when we now sample these triplets, we are doing it in what we call a batch-aware manner. So for example, we have 2 studies with 2 cell types. And when we select an anchor point, we make sure that the positive pair is the same cell or a cell that's very similar transcriptionally, but it has to come from a different batch, from a different study. And then the negative, that's a different cell from this first study. And so this way you can actually define a bunch of triplets. And as you define these triplets, the anchor and the negative will always be from the same study, and the anchor positive will be from different studies. And one of the advantages of this technique compared to other approaches is that you can actually generate almost an unlimited amount of data points. Just 20 samples here, they will give you 500 unique triplets. So the question is, does this work? And so we first applied this algorithm to simulated single-cell sequencing data, and we simulate it in such a way that we have strong batch effects. So what you can see here is that essentially each batch and cell type combination is a separate cluster. When you look in the right-hand side, in red, that's supposed to be the same cell type, but there's 3 clusters because the batch effect is dominating this embedding. So the question is, what happens when we apply our method to this data set? Now what you will see in this animation is that the longer the algorithm trains, the more it understands that there's sort of these different clusters, and it actually manages to find an embedding where batch is sort of taken out, but the grouping information remains the same. So that coordinate system, in a way, is more meaningful than the coordinate system we had in the beginning. We can also apply this to real data. So again, this is the example of the pancreas data from 4 different studies. And as you can see, when we apply our method, you can look at the right side, the colors sort of match better. And that suggests that we come up with an embedding or a representation that kind of ignores batch and gives you more of a biological view onto the data. So of course, there's multiple methods that do that. I think it was sort of the challenge of these last years, how to best integrate, and every method has their pros and cons. I think our method here has an advantage that it actually allows you to scale pretty well. So we integrated more than 2 million mouse brain cells from 4 different resources. And actually, in terms of speed and RAM usage, our algorithm is pretty user-friendly. So you can integrate over 2 million cells in an hour and a half on a standard computer. So you don't need a high-performance computing system. And I think that really empowers sort of smaller researchers or smaller groups, maybe without the capability to have access to high-performance computing systems, to perform integration at the Atlas scale level. And so, with that, I would kind of like to allude to a historical moment in molecular biology here that, depending on your perspective, the data and the biology and your view of it can really change. And I think this is sort of the main takeaway message I'm trying to convey, that when you look at single cell embeddings, you need to always be aware of what's the right background, what's the coordinate system, and really think if you're asking the right question by projecting them into the right space. So with that, I would like to thank you guys for your attention. And I'd also like to thank the people who helped me contribute to this work, specifically from UT Health, Zhongming Zhao, Yinying Wang, and Fangfang Yang. Thank you.
Thank you very much, Lucas. This was a great talk, and I think we have about 5 minutes. And again, if anyone has questions, please ask them on Umbrella. And we have a question here from Dan Truong. And the question is, does insect, I don't know, this is how you pronounce it, insect, adjust gene expression data similar to Seurat integration? Or is it similar to BBKNN and Harmony, where you only generate an embedding?
Yeah, that's a very good question. And the short answer is it only generates an embedding. So it doesn't give you kind of correct the gene expression data. But we are trying to explore ways to include that in our method. Good question.
Okay, so I have a question here. So when you look at these kind of views and different coordinate systems and so on, how do we know which one is the true? I mean, as you said, there are multiple views. So it's not that one of them is true and the others are wrong. But how do we know that what we are getting from these methods is correct?
Yeah, I think that's a very good question. And I get it asked, I think, many times. People hunt sort of for the true embedding. Usually at the very beginning of some analysis, they hunt for the true embedding, and they want to sort of generate one embedding and then be done with it and base all of the analysis on this one embedding. But I think that there is no single truth, and it really depends on the question that you're asking. So depending on what question you're asking, that is sort of the perspective that you should take on the data, right? For example, if you're interested in sort of cell cycle state, maybe you would include cell cycle genes in the calculation of the embedding or so on. If you're just interested in cell types, differentiating cell types, then you would use a method that's specific for that. So I think that when we think about these data manifolds, we need to really appreciate the ability that there are many different perspectives to look at the data that can give you answers to different questions. And I think that is sort of one of the beauty of single cell. It's so complex, you can ask many questions of the same data set.
Thank you. One more question from Courtney Hodges, who says, fantastic talk. Are there criteria that can help prevent overfitting with imputation, similar to Bayes information criteria or Akaike information criteria?
Right, so that's actually a very good point that we should always be aware of. That when we're when we are denoising the data, we're essentially introducing some sort of additional correlation structure on top that may not necessarily be true biology. And it's really tough to actually know where to cut off and where not to cut off. So to answer this question, in particular in our paper, we try to show that when you that we have some sort of buffer against overfitting, and we showed that if you look at genes, and we did this in simulated data, but if you look at genes that are not differentially expressed between two types, they should not have sort of any clustering structure. And I think we have a supplemental figure on that that shows that only genes that were truly differentially expressed in simulated data, only those really separate the clusters, and those that we simulate to have no effect of differential expression, those didn't restore the clustering structure.
Okay, we have less than a minute, and we have one last question. I will ask it here by Hyun Hwang Jeong. Is it possible to use INSEC to embed multiple data sets if each data set has its own batch effect?
Yes. So the concept is built on the assumption that each data set has their own batch effect, but there are some kind of additional assumptions that we make that have to be given. And I think one of the assumptions is that at least one type of cell needs to be present in both batches. So if you're looking at two batches that actually have no cell types in common, then you could run into problems. But if that assumption is given, then Insight should be able to give you an embedding that's more meaningful than without a batch.
And with that, thank you so much, Lucas. This was a great talk, great answers for the questions. And we now come to the last, but definitely not least, speaker for the day is Dr. James Kai. He's an associate professor of computational systems biology in the Department of Veterinary Integrative Biosciences at Texas A&M University. He is also affiliated with the Department of Electrical and Computer Engineering there at A&M. Dr. Kai's research lies at the interface of human genetics, computational statistics, and data science. And with that, James, please take it.
Thanks for the introduction, and it's my great pleasure to be invited to present in our recent work. Revolution is an overused word, but I think it's difficult to overemphasize here. We are at the intersection of two major technology revolutions. One is the single cell technology, another is machine learning. So the single cell revolution started a couple of years ago, gave our capacity to measure gene expression level in thousands of cells and the whole transcriptome. This provides a challenge to data science as well. So if you look at this picture, the y-axis is the expression level about a lot of genes in a couple of thousands of the individual cells. So those cells are homogeneous population. There are no difference in their cell types, but still you see a lot of variability among different cells. So this gave our a new perspective among the between cell variability. So traditional differential expression gene compared to the mean will not work sometimes here. And also the data set normally is a high dimensional nonlinear system. So require machine learning algorithms such as manifold learning to learn the structure of the whole global structure of the data points. So we will touch on base of those applications of those. So my lab has been working on to build so-called MATLAB toolbox and to linearize those selection of different algorithms. And from this overall picture, you can see normally you can do embedding, dimension reduction. You can do trajectory analysis. But the main feature of these solutions in our pipeline is we can build single cell gene regulatory network. And we can build this along the pseudo time series, you end up with a series of the time series network, and this automatically becomes the tensor structure. And you can learn the dynamics about this time series, so-called pseudo time series regulatory network. And then we also emphasize on the usability of this picture. So I will show you this video, if I can play it easily. I think it's very important to have good interaction tool, because browser-based gene browser of this single-cell RNA-seq analysis just not user-friendly. Sometimes you need this high-level working environment to have interaction between the end user and the data points you have. Here, I'm showing you a cell type annotation tool. You can just drag and see what the cell type they are. This is a particularly a sample from a pancreatic cells. So you see alpha, beta cells, gamma cells in there. Okay, so we move forward and try to build some more sophisticated solutions as well. So I'm going to mention a couple of machine learning tools we built recently. You can see the name of these three packages all start from single cell tenifold. So those are the reason is because they all based on the tensor decomposition and the manifold alignment. So we gave this some similar name. Today I'm going to focus on the first two, which we can use this to construct and compare single cell gene regulatory networks. And we also have this knockout tool. We can do virtual knockout and also the cell-to-cell interaction study. And this paper has been recently published in Python, a new journal for data science. And the source code is available at GitHub here. Okay, I want to go back and see why we are interested in single-cell gene regulatory network and why we can use machine learning tool to review the meaningful expression program in there. So we have sequenced a couple strings of lymphoblastoid cell lines recently, and we construct co-expression network and found a lot of those highly variable genes also form tightly related network modules. So this gave our intuition that we probably can learn the network, although they are gene expression from different cells, but we learn from the cell population can infer what happened in the gene expression program. So this is a well-known regulatory network module, and it's not from a single cell itself, it's from an experiment, most of them from knockout mouse. This is an NF-kappa B regulatory network for the active B cell, and also this B cell line has been studied very well, and people come up with this model, and you see this kind of model, which gene regulatory network have been in different scenarios. So we ask whether we can recover the correlation between, for example, these two genes, ERL positively regulate AID, and look at our lymphoblastoid cell line. Lymphoblastoid cell line is derived from the B cell. It still have the features of a B cell, and these two genes is positively correlated among cells. So each point here is a cell, and the correlation coefficient is really high, indicate this is can recover what we know from the known model of the gene regulatory relationship. And then we look at all pairs of the genes correlation between all the members of those genes. And this matrix is symmetric, so you may only focus on one side of the matrix is okay. And if the correlation between this pair of genes can be recovered correctly, I mark it as a red box. And you see this is the red box will be in this. And only 2 of this correlation has not been correctly predicted from the single cell gene expression, co-expression correlation. This is negative, but based on the model should be positive. And also, if you focus on this, is correctly predict these 2 genes correlation. But if you think about the 3rd gene, so these 2 genes should be positively correlated among cells if the cell has this 3rd gene have a high level expression. So this color bar indicates the gene expression level of BL and P1. And if this 3rd gene have a lower expression among, for example, this group of cells, the correlation between these 2 genes become negative. So clearly, there's a nonlinear relationship among the correlations between them. So it's not possible to use only pairwise comparison to do this analysis. So we have to think about the networks. So also, constructing network is one issue. How to compare them once you have a single cell gene regulatory network from condition 1, from condition 2. How are you going to compare them? For example, I want to identify a gene, like gene number 2, have a rewiring event happened. It was strongly correlated with 3 other genes, but once the condition changed, gene 2 will not be able to maintain the same kind of relationship pattern with other genes. So that's the goal we are going to design and implement the algorithm here. So the algorithm we implement is called the single cell tenfold net, and it's a machine learning algorithm with multiple steps. So this is an overview of this workflow. And I will go through the main steps one by one. And we start from 2 data sets from single cell. So you can think about this as one wild type, one is a knockout, or one is healthy control, one is a diseased sample, whatever. The first step, we use cell subsampling. So we want to control for some of these outlier cells to have an impact on our network construction. So we subsample those and make a couple of subsample data sets. And for each of these data sets, we construct single cell gene regulatory network. Okay, each of these constructs one of the network is present by the adjacency matrix here. And the approach we use is called the PC regression. It's underused approach. Nobody has ever used it for single cell RNA-seq analysis. It stands for principal components regression. And we found it's pretty good. And using simulated data, we can conform is beat all other available machine learning or other algorithms. So this is only shows 3 of them, but in our paper, we compared with 11 other software package people have been used for the single cell gene regulatory network construction, but the PC regression really standing out. Okay, the next step, once we finish the construction of the regulatory network for each subsampled data sets, it forms a tensor, and we use a tensor decomposition to decompose all this, try to use several one-order components to learn the structure of those. And then we reconstruct using top components. The purpose of this is to denoise, okay? So tensor decomposition and this approximation has been used widely in machine learning field for people to process image or video data. And for example, you can denoise those video, and also you can extract latent information out of it. So the same idea as matrix decomposition, but it's just higher dimension. And you can select how much noise you want to filter out. If you only use 3 top components to reconstruct your network, you will reconstruct the major signals. So this is one of the regulatory network adjacent matrix. But if you use top 10 instead of 3, you will reconstruct the whole network in more detail. And this if the noisy network, you have more noise in there. So this is some just visualization, and those are the subsampled networks, and before and after the noise, and then you take average. So once you have done this, these two denoised matrix will be ready to compare to each other, because all the noise signal has been filtered out, and anything different between them will be highly significant biologically, hopefully. And the last step, we use manifold alignment to align those two denoised network and put the same gene in the same manifold structure, and then look at the relative position of their before and after treatment in the two conditions. And then, depend on the distance, we can rank gene. Okay, the outcome of this will be a list of ranked gene from the analysis that will reflect the difference between these two sets. So that's the definition of the manifold. Is abstract and mathematical space, and their feature is you have a local Euclidean distance available, but not valid when you look at the more distantly related data points. As I said, the end point of this pipeline is a list of genes. It's ranked by their distance on the manifold. And then you can do the downstream enrichment analysis, what kind of functional association about those gene sets. The gene we call differentially regulated gene. Okay, so this can be used as a complement test to replace differential expression analysis. The signal it recovered is not the same as the differential expression analysis. As I said, we want to recover the gene. Those genes will be on the top. They have a dysregulation or differential regulation between these two networks. So we test our pipeline using a lot of pre-published data. So those data have been published, and people use differential expression to understand what's going on before and after treatment. So they all have two samples to compare. And this from neuron, and this is from a cancer cell line. This is a lung cell epithelium, fibroblast neurons, and their treatment and stimulated Y type, etc., etc. So we try to see what we can learn from them. The first example I want to show you is the data generated from a mouse and treat with a morphine. And the original paper didn't find anything in the neurons, although they designed to find what's the morphine impact on this mouse neuron. So they have to change the title probably to the glia, any cells other than neuron. So they couldn't find anything really. It's unsurprised that if you look at the gene expression of this morphine response gene between before and after treatment, there's no difference at all. And overall, this before and after treatment, there's no difference as well. What you can found is using our pipeline to find this ranked gene, and top ones you found morphine addiction gene set and the opium and the signals. So all makes sense. And also this is a local network contains the most substantial regulatory change between this central model of the regulatory network before and after treatment. Similarly, I will quickly go through those, and this is a anti-cancer drug and apply to the cell line, and also we found all the mechanisms which people know how this drug will work. And this is from another example from the knockout example, wild type mouse against the knockout, so we can predict what's going on here. This is a stimuli and the double-strand DNA as a stimulate to the apply to fibroblasts, and also we recover what we expect to see and from this stimulation. And the last one is a comparison between wild type mouse against Alzheimer's disease model mouse, and we recover the major related gene network change, and that's the module change, and the gene sets, everything makes sense. So this is perfect. So the last section, I will apply this to COVID-19 single-cell RNA-seq data and see what we can get. The raw data we get it from this paper, and there's people sequenced the macrophages from a broken alveolar immune cells. So we download the data and visualize it, and there are two groups. One is from patients with mild or moderate affected symptoms, and also the group with the most severe conditions. So it's a mildly affected with a severe condition patient comparison. The data from either macrophage or dendritic cells, we can use marker genes to identify those cells. Apply this, we have a 27 highly significant differential regulated gene has been fished out, and that's top genes. Then we look at the gene set enrichment analysis using the whole list. Remember, we have a ranked list, and unsurprisingly, and the top pathway is the type 1 interferon, and this immune response related. But the most interesting gene set we identified is malaria and the phagocytome. Okay, and those are the example genes which pop up associated with these two pathways. We think is more important. Remember, we are not detecting any differential expression. It's a regulatory change. So some gene will be upregulated, some gene will be downregulated. So, but as a whole pathway of the malaria infection pathway in the KIG database, we can see some of these key genes have been upregulated, some have been downregulated compared between what mildly between patients with mild symptoms with more severe condition patients. So our argument here is, although there's intensive debate over whether we can use anti-malaria drug, hydroxychloroquine, for example, these drugs have been used as anti-malaria for more than 60 years, probably. And our analysis, which there's no political issues, purely data science driving discovery, we found the patients between using single cell data is differed by malaria pathway in their regulation of the gene in the single cell in their immune cells, and also the phagocytome pathway as well. If you look at the review paper, how hydroxychloroquine will work within, yeah,
Two
at the
minutes.
single cell level, and it will inhibit immune activity through this cytokine pathway downregulation, and also in fact the lysosome activity. Okay, so that's, if you match this, and the implication here is we discovered this major difference between these two groups of patients, and the gene expression level difference match perfectly with the hydroxychloroquine suggests the mechanism of how this the drug will work. So we suggest that probably we can use this, and we just maybe need more thorough clinical studies to see whether we can use this for treatment for the mildly affected individuals to prevent them to develop more severe conditions. And this has been released as a preprint in this URL, if you like. And at the end, I will acknowledge that Daniel Osoro, and he a wonderful graduate student, and lead this study. We have collaborators from statistics department, Professor Huang and Julian and Guanxun. With that, I'm ready for any questions.
Thank you very much. Very nice talk. And we are basically out of time, but there is one question I will ask it here. So for your COVID data, the Liao et al., what did those DEGs refer to? In which two conditions?
The two conditions is one is, so that study had three group of one healthy control and the two conditions. One is mildly affected, another more severely. So from the clinical side, I rely on the published data. I didn't do any research. I just grouped them by the data they provided.
Okay, with that, I want to thank James, and I want to thank Ruli and Lucas for very, very nice talks. And I want to now hand it off to Ray for concluding remarks.
Thank you, Louis. So before we conclude the meeting, I'd like to take the opportunity to thank the speakers for the wonderful talks, and also our poster presenters for their posters. And also thank you for the participation. And also, I'd like to thank the organizer and moderator, Nick, Louis, Hongyu, and Zhongming, for really keeping us on time. The consortium, the cluster, and then this meeting is sponsored by UIS and GCC, and also, this meeting are sponsored by the company you already know, 10X, Illumina, and then Protein Simple. Thank you for the support. This is really the first time, we have the GCC online meeting. So it's really very smooth meeting, I would say. And then this is not possible without the GCC staff, Suzanne, Dawn, and Kara. Thank you very much for your fabulous work. I do want to note that there's a survey. There'll be a survey sent out to you. So we would like to hear back from you for the feedback on the meeting. And also, since our mission for the cluster is try to stimulate collaboration, education, things like that. And then we'd like to hear from you about what we can do to do it better to help the community. So feel free to email us. You can visit our cluster website, and there's a contact information. Just feel free to email. So with that, I'd like to thank you guys again, and have a nice rest of the day. Bye-bye.